Okay, so lecture 4.2. Lecture 4.2, we're not going to be looking at the internal structures of eukaryotic cells. Okay, here's where we're going to blast through. Okay, you've seen this, you've heard this stuff, so I'm not going to spend too much time. Okay, first thing we got to focus on is what defines all cells as eukaryotic, the fact that they have a nucleus. Okay, the nucleus, it's a dual membraned organelle. Okay, it's something that's housing stuff that's so important, the DNA, that evolution has gone about wrapping it twice. You have two lipid bilayers. To get in and out, you have those nuclear pores. It's going to limit what can and cannot get in and out. The chromatin, which is our DNA, the double helix that you've heard of since you were a small child. That DNA is wrapped around proteins, and that DNA-wrapped protein is known as chromatin. The chromatin will then wrap around itself, and you get larger and larger structures of it wrapping around itself, and other proteins, you get what you refer to as chromosomes. Important thing to remember is that the nuclear envelope, that dual membrane, is actually continuous with the endoplasmic reticulum. So that messenger RNA that leaves through the nuclear pore immediately runs into the endoplasmic reticulum. Endoplasmic reticulum, okay, comes in two forms: smooth, rough. Smooth, its job is making new lipids. Its job is detoxifying anything that gets inside the cell. Turns out, everything you eat has a little bit of toxicity, a little bit of poison in it, either directly in it or as you're metabolizing it, some of the byproducts, the waste products. So Smooth ER takes that, gives that compound a little twist, a little tweak, whatever it takes, change its shape, change the function. What was toxic, was poisonous, no longer is. Rough ER, rough because it has studied with ribosomes. Ribosomes will make polypeptides, the polypeptides that will fold in on themselves and combine with other polypeptides to form proteins. And then you look and you see the Golgi. Okay, the Golgi is another organelle that looks like stacks of pancakes. It's ribbons and ribbons of membranes. Each of the little foldings has a slightly different environment. Some are going to be more acidic, some are going to be more basic, some are going to have this enzyme, that enzyme, lots of sugars here, lots of amino acids there. It's whatever is needed to get that final structure, that final format for whatever protein, glycoprotein, lipoglycidic, whatever, that needs to be made by the cell. This is where it's formed. So when you look at this figure above my head, and you follow the pathway, you will see messenger RNA, the copy of a gene from the chromatin, the chromosome. That messenger RNA is going to leave out through a nuclear pore. First thing it's going to run to, rough ER. Ribosomes are going to read that messenger RNA, make a polypeptide. That polypeptide is going to be trafficked through the different places in the ER, the smooth, the rough. A little twist, a little tweak here and there. Something added, something taken away. Then it's going to be put into a bubble, a lipid bubble that's part of the ER that gets torn away. They call that a vesicle. Vesicle is going to take the proteins, take the new whatever, and it's going to transfer it to the Golgi. Once it's in the Golgi, it's going to go through all the different areas. Final twists, tweaks, sugars added, sugars taken away, combined with other polypeptides to form mature protein complexes. And then from there, it's going to get into an even larger vesicle and go to the cytoplasmic membrane, where it's going to fuse and then be emptied out into the area around the cell. Now, what's interesting is that whole process I just talked about can be done in reverse under certain circumstances. You look up above my head at step 1. At step 1, you see what's called the food particle. It comes in contact with the cell. Food particle could be a piece of another cell that died and it now needs to be removed. It could be a bacteria that got into your gut, a virus. Anything smaller than the cell itself now becomes food. It's going to engulf and wrap around it. As it's wrapping around it, part of the plasma membrane is going to pull off, and now is going to be a large membrane bubble inside the cell. They call that a vacuole. If it's small, vesicle. If it's big, it's a vacuole. That vacuole is going to fuse with other vesicles that are storage vesicles. These storage vesicles have digestive enzymes and stuff in them. They call these storage vesicles with digestive enzymes, they call these lysosomes. Lyso, meaning lysing. So literally, as you see through steps 5, 3, and 4, what was a food particle gets broken down. Peptides, the proteins, get digested down to amino acids. Anything that's, polysaccharide-like gets digested down to individual sugars. That cell now has something to eat. Another organelle that has absolutely nothing to do with the ER, the Golgi. Mitochondria. Mitochondria, you've heard it. Mitochondria, powerhouse of the cell. And that's literally what they do. They are the site for oxidative phosphorylation, the making of the majority of the ATP that the cells are going to use as their energy source. It's not the only thing the mitochondria do. The mitochondria also interact with the rest of the cell and help, maintain the cell, help with regulating the cell, also help with signaling of when it's time to die. Cell apoptosis. All cells have the ability to commit suicide, if and when those signals come through. And then finally, you'll see chloroplasts. I'm not going to talk too much about chloroplasts because for most of the microbes that we will talk about in this course, being able to do photosynthesis really doesn't have much to do with us. Think about it. Once they're inside of us, no sunlight, they die very quickly. So they really can't, for all intents and purposes, they can't cause us an infection. Okay, now you may get sick if you drink, some pond water that's full of scum, and that scum is photosynthetic algae and protozoa. Yeah, that's an ingestion, not an infection. The ribosome, cytoskeleton, I'll let you read on those.